Hello, my name is Johnny Blazeck, and I'm an assistant professor at Georgia Tech. And I was awarded a Beckman Investigator Award in 2020. And I'd like to talk a little bit about my research journey and how the Beckman Investigator Award really ties together everything I've learned from sort of my first time going into a laboratory towards where I hope my research program keeps going in the future to help society. I'd also like to talk about how the life and work of Dr. Beckman, shown here receiving the National Medal of Technology in 1989 from President George H.W. Bush, but how his life and work has really helped my lab's work, my scientific career, and my scientific journey. So Dr. Beckman is, of course, considered one of the greatest inventors of scientific instruments of all time. And my lab, kind of on a daily basis, uses a lot of the devices that he created, which really helped revolutionize the study and the understanding of human biology. So shown here is the spectrophotometer and the pH meter with newer versions included into the kind of additional versions. Um, and Dr. Beckman also sort of showed the power and the importance of developing new inventions to help scientists do better work and do more work faster. But I've also tried to emulate Dr. Beckman's vision in terms of really trying to help the human condition and also making sure that my lab's work is of the highest quality. And before I get too far, of course, I'd like to give a big thank you to the Arnold and Mabel Beckman Foundation for their generous support. I was fortunate enough to receive a Beckman Investigator Award early in my independent career, and it's really been a catalyst for almost everything that we've been able to do since then. And to explain the work that we focused on with the support of the Arnold and Mabel Beckman Foundation, I'd like to tell you a little bit about my research journey in terms of how I got here and why we pursued developing the invention that we were interested in while I was a Beckman investigator and where I think this can kind of continue to take us in the future. So going back many years, I was always interested in engineering cells to be able to have new or better functions. So as a PhD student, I worked with a type of cell called a microbial cell, or more specifically, a type of cell called a yeast cell. And what I was working on was taking these yeast cells and trying to use them as sort of cellular biofactories to convert waste from farms into biofuels. And this was a lot of fun, and we could engineer these cells like kind of very detailedly in many different ways, and to the fact that they went from kind of shown here on the left, the way they started, towards being almost completely full of these biofuels. Now, while I liked engineering cells generally, I also wanted my work to have more kind of direct applications in biomedicine, or more specifically to be able to improve human health. But fortunately, I was able to sort of switch gears with the next research that I did and start studying something called immunoengineering. This really kind of continued to help set the stage for the work I did with the Arnold and Mabel Beckman Foundation. In this field of immunoengineering, it really just means trying to make an immune system or somebody's immune system fight a disease better. And if you've heard of the immune system, you may have heard of things called immune cells, like B cells, which are the cells that make antibodies that are so important for fighting off a lot of diseases. And there's also another cell type called T cells, which are very important for fighting things like viruses and cancer. And during this project, I was working with a protein that we were using to try to make T cells work better. Now, unfortunately, this protein wasn't very strong. It's shown here. It's kind of its molecular structure, and all these sort of red and orange dots are the different things that we tried to sort of make it better. And it turned out the bottleneck was often kind of making a lot of different changes to the protein and then testing them in mass. And this kind of took years of tedious work to really get anywhere. And it turns out that while this protein was supposed to help T cells, that B cells could sort of have the answer for this issue, where B cells, they can actually mutate antibodies to make them better kind of constantly. And they do this continuously to help body fight off new diseases. And while they're not exactly the same, the protein I was working with in antibodies, they have the same general features in terms of they can be used to help the immune system, and they both come from DNA. So I was really interested in trying to use what B cells do to be able to make antibodies and make proteins better. Now, unfortunately, B cells, they're very fragile. They're not really designed to be out of the body. But I remembered from my prior work that that yeast cells are actually, you can do a lot of things to them in terms of engineering them. So my goal as a Beckman investigator was really to take all the machinery and all the tools that B cells use and put them into yeast cells so that they can kind of really robustly and rapidly make new immunoproteins like antibodies. And it took a lot of kind of complex cell engineering where we were working with kind of other inventions like the recently Nobel Prize awarded CRISPR gene targeting system, and also things like base editors that kind of can help mutate DNA and engineer proteins. But it all kind of came together to the fact that we're able to kind of, one, have yeast cells have antibodies inside them, and then two, to be able to kind of constantly have yeast cells make these antibody proteins better in the exact same way that B cells do. And actually to be able to do this kind of faster than anybody's ever been able to make proteins better before. So with this sort of technology, we could kind of make a lot of different types of antibodies better, or a lot of different proteins better, including antibodies that can actually be used to target cancer. And this sort of ability to really quickly engineer proteins or engineer antibodies using yeast has been really important towards my lab as we kind of start to really try to go after a lot of impactful sort of immune system engineering problems. And we think it could be really helpful towards engineering kind of other immune cells to be able to fight cancer better. For instance, that other immune cell type T cells, having them secrete proteins that can fight cancers or fight diseases, and then also engineering other proteins like enzymes themselves. So with that, so to conclude, I'd also just like to say thank you to the Arnold and Mabel Beckman Foundation one last time for their generous support. And I'd also like to say that I kind of fully expect to see new inventions and new technologies coming out from other research that being supported by the Beckman Investigator Award Program, because I do think that it's sort of the best system to get new impactful inventions that help out help the human condition in general. So with that, thank you again, and bye.